Hi and welcome back to another A-Level Biology video with Starfish Revision. This one is all about proteins. We're going to cover the structure of an amino acid, how amino acids join together, how a protein folds up into its final structure, and of course we're going to look at the food test for proteins. So before we start, can you think of any biological uses for proteins? For example, enzymes are proteins. You can pause the video and then I'll go through the list. Right, so examples of proteins are enzymes, antibodies, transporter proteins, receptors, some hormones are proteins, hemoglobin is a protein, and there are structural proteins such as collagen and keratin. Collagen in the skin, keratin in the hair and the nails. Well done if you got any of those. Proteins are long chains of amino acids. An amino acid has this structure: a carbon in the middle with an amino group containing nitrogen on one side and a carboxylic acid on the other. There's one H group on the bottom, and then there are 20 different amino acids that exist, and that's because each one has a different R group at the top. It's called the R group or the variable group, and examples are glycine, where the R group is just another hydrogen, alanine, which has CH3, and arginine has, well, a different one again. Remember, it's only this R group that differs between amino acids. The rest of the molecule stays the same. Now, two amino acids join together to form a dipeptide. It's a condensation reaction, which means a molecule of water is removed, and the bond that is formed is called a peptide bond. This reaction can be reversed in a hydrolysis reaction, for example, during digestion. So we've made our dipeptide, but a whole protein is hundreds, perhaps thousands of these amino acids joined together in a really long chain. So that's hundreds or thousands of condensation reactions joining them together. These polypeptide chains must have the right shape to perform their function. And the way they get into that shape is by folding themselves up. Bonds form between different amino acids at different points of the chain. Because the amino acids are different, they interact with each other in different ways. Amino acids are first linked together in a particular order. We call the amino acid sequence of a protein its primary structure. Depending on the order of the amino acids, sections of the polypeptide chain arrange themselves by forming hydrogen bonds with neighbouring amino acids in the chain. This results in either a spiral arrangement called an alpha helix or a zigzag arrangement called a beta pleated sheet. We call this first bit of folding a protein's secondary structure. But the folding isn't finished yet. The polypeptide chain then folds itself into its unique 3D shape. This is the protein's tertiary structure. And it happens because more bonds form between different amino acids in the chain. These bonds may be hydrogen bonds, ionic bonds, or disulfide bridges. Once all this folding has taken place, the finished polypeptide has been made. But sometimes proteins contain more than one polypeptide chain joined together. Hemoglobin is an example that contains 4 polypeptide chains. If this is the case, we say the protein has a quaternary structure. You will sometimes see a protein represented by a diagram like this. You can see the alpha helix sections and the beta pleated sheets are represented by flat arrows. So, to summarise, primary structure is the sequence of amino acids making up the polypeptide chain. Secondary structure is the initial folding of the chain. Some sections will fold into an alpha helix and some sections will fold into a beta pleated sheet. Tertiary structure is the further folding of the chain into its 3D unique shape. And quaternary structure is when more than one polypeptide chain joins together to make a bigger protein. Now, what is the food test for protein? Well, it's the Bioret test and it has 2 steps. Step 1. Add a few drops of sodium hydroxide solution to the test sample to make it alkaline. Step 2. Add some copper 2 sulfate solution. If protein is present, this will turn purple. If not, it will stay blue. So that's it for proteins. Hope you found it helpful. Please like and subscribe if you did, and I'll see you in the next video. Thank you for watching.